Good morning everyone. So let's today discuss about the structure of the genetic material, that is DNA. Now while we are studying about the D structure, so how this structure was observed. So basically there are two investigations which was done. One is called the Chargaff's rule and other is the X-ray crystallography. According to Chargaff's rule, which was formulated by Erwin Chargaff, he said that the amount of purine is always equal to that of the amount of pyrimidine in a DNA structure. Okay, that means A plus G is equal to T plus C. And also he stated that the amount of adenine is equal to that of thymine and that of guanine is equal to cytosine, because we know that while they are forming complementary base pair, the amount of adenine and thymine has to be equal, isn't it? So this is as per the Chargaff's rule. Then the second investigation is the X-ray crystallography. Here this was done by Maurice Wilkins and Rosalind Franklin. They observed that during the crystallography study, the X-ray diffraction studies, pictures that were taken, they observed a three-dimensional structure of DNA which consisted of two polynucleotide chains which were about 20 Armstrong diameter. One complete turn consisted of about 10 base pairs. The length was 34 Armstrong and so the base pairs distance was about 3.4 Armstrong. Now, based on this study, Watson and Crick proposed the double helical structure of DNA. Okay, now this double helical structure, it consists of two polynucleotide chains, glycosidic and phosphodiester bonds, the DNA duplex. So now gradually let us discuss each and everything regarding the DNA structure. Okay, so first of all, okay, now while we are discussing about the two polynucleotide chains, so poly we know that means many, nucleotide. Now what is actually nucleotide? It consists of nitrogenous base, okay, then pentose sugar and phosphate group. Now we know that if nitrogenous base and pentose sugar is present, then we call it as nucleoside, and if phosphate is present, then it is a nucleotide. Now when nitrogenous base, that means here two type of nitrogenous base, one is purine, other is pyrimidine, isn't it? Now purine, again it is of two types, adenine and guanine, pyrimidine, cytosine and thymine, and pentose sugar we know it is deoxyribose in case of DNA. Now here in the nucleotide, the nitrogenous base is attached to the sugar and the phosphate molecule. Okay. Now here the purine adenine is bound to, or it forms complementary base with that of thymine and guanine with that of cytosine. Now adenine and thymine pairs with the help of two hydrogen bonds and guanine and cytosine with the help of three hydrogen bonds as per their chemical structure. Now purine consists of two rings. It is a two-ring structure and pyrimidine is a single ring structure. All these we have actually discussed in the last class, isn't it? So now we know all these things. So let us now draw the structure of the DNA molecule. Now the DNA molecule, now here you see this is a new polynucleotide chain. Again this is the other polynucleotide chain. Now the DNA molecule, the polynucleotide chain, it consists of deoxyribonucleotides, okay, which is formed of the phosphoric acid, the deoxyribose sugar and the nitrogenous base. Now there are, we know that four nitrogenous base, adenine, guanine, thymine, cytosine, that means purine and pyrimidine, isn't it? Now here the four kinds of deoxyribonucleotides are formed actually. Deoxyadenosine 5 monophosphate, deoxyguanosine 5 monophosphate, simultaneously deoxythymidine 5 monophosphate, deoxycytidine 5 monophosphate. Now these are actually the different type of deoxyribonucleotides that are formed. Now you see that the nitrogenous bases are arranged towards the central position, okay, and the sugar and the phosphate, they form the what is called the backbone of the main nucleotides. Now here actually the glycosidic and phosphodiester bonds are formed. Glycosidic bond, that is the nitrogenous bases which are attached to the sugars with the help of glycosidic bond. That means these are the nitrogenous bases, they are attached to the sugar molecule with the help of glycosidic bond. Again, these sugar molecules, two sugar molecules are attached to a phosphate molecule or a phosphoric acid molecule. So this is the phosphodiester bond. Got it? So this is the phosphodiester bond, this is the glycosidic bond, and the two polynucleotide chains, they are held together with the help of the hydrogen bonds. Now they remain spirally coiled, okay, and this duplex actually it is of about 20 Armstrong in diameter. Okay, now you see that these are the two polynucleotide chains which are spirally coiled. Okay, the coiling becomes plectonemic, that is the two strands, they cannot be separated without completely unwinding. That means we have to open it. If we don't open it, if we don't unwind it, we cannot separate the two strands. Okay, they remain very closely packed together. Now this diameter is about 20 Armstrong. Now so you may be thinking that how come that throughout the whole length it is 20 Armstrong. Actually what happens, we are repeatedly telling that a purine binds with pyrimidine, simultaneously pyrine purine, isn't it? So purine, it is a two-ring structure. Pyrimidine, that is a single ring structure. So this is suppose adenine, it will bind with thiamine, which is a pyrimidine. So what happened? Three ring is formed. Again, cytosine, single ring structure. Guanine, double ring structure. GC will pair, isn't it? Three ring structure. That means this three ring constantly runs throughout the length of the DNA. That's why the diameter remain constant, which is 20 Armstrong. Now this is one complete turn. This complete turn consists of about 10 base pairs. Okay, this is actually the normal structure of DNA, which is also called the B form of DNA. It consists of 10 base pairs in one turn, and this length is about 34 Armstrong. That's why the distance between each base pair will be about 3.4 Armstrong. Now these are actually the basic things which are present in a DNA molecule. Okay, now the deoxyribose and the phosphoric acid, they form the backbone. This is the backbone, so they are forming the backbone, while the nitrogenous base, they remain at right angle to one another. Okay, now this backbone is formed of alternative deoxyribose sugar and phosphoric acid, and the polynucleotide chains, they show polarity. Polarity means here if one end is 3 prime, the other will be 5 prime. Similarly, this will be 5 prime, this will be 3 prime. That means they show polarity plus they are antiparallel. Okay, one end is if 3 prime, the other will be 5 prime. So they are antiparallel in throughout the length. Now they show complementary base pair. That means if one strand will have adenine, the other strand will have to contain thiamine. Okay, so that is the complementary base pairing. So therefore this obeys the Chargaff's rule, because if adenine is there, thiamine is there, that means the number of adenine is equal to that of thiamine, and simultaneously the number of guanine will be equal to that of cytosine. So these are according to the Chargaff's rule. So this is the basic actually structure of the whole DNA. Now again a question will arise, what is actually 3 prime and 5 prime? Now we are telling that the sugar is pentose sugar, that means 5 carbon. Okay, if the phosphoric acid is attached to the third carbon position, then that is known as the 3 prime end. Okay, and if it is attached to the 5 carbon position, then it is known as the 5 prime end. That means if one attaches to the 3 carbon position, then simultaneously it will attach to the other sugar molecule at the 5 carbon position. That's why if one end is 3 prime, the other becomes 5 prime. Okay, So the 20 Armstrong concept and 3' 5', what is it actually? These two are important things. These are basic concepts which you have to know while studying the DNA structure. Okay, so this is all about the DNA structure, different type of DNAs we found. So we are now studying about, this is the basic B form of DNA. Okay, so while we are studying the structure, what we have got? First of all, we have got the Chargaff's rule, X-ray diffraction, crystallography method, then Watson and Crick's double helical structure. Now these two are the polynucleotide chains which consist of nitrogenous base, phosphoric acid and pentose sugar. They combine together to form the nucleotides, and the nitrogenous base remain towards the inner side, phosphoric acid and sugar towards forms the backbone. The nitrogenous bases are attached with the help of hydrogen bonds, and the nitrogenous base and sugar are attached with the help of glycosidic bond, and the sugars with phosphoric acid forms the phosphodiester bond. Okay. The two strands remain spirally coiled. The diameter is 20 Armstrong. The length of one turn is 34 Armstrong. Each turn consists of 10 base pairs. So the distance between each base pair will be 3.4 Armstrong. Now they show polarity. If one end is 3', the other will be 5'. They run in anti-parallel direction and they show complementary base pairs. Okay, so these are all the basic things regarding the DNA structure. Go through the video properly, go through the notes, study them. If you have any problem, then let me know.